For a whole month, or?
Just this week.
Oh.
Moving to Illinois.
Uh-huh.
Yeah, so got to get back and pack up. Okay, we're going to start the afternoon session with Richard Durbin. There's this surprise title: Girls Welcome Transform-Based Methods for Assembling Haplotype Analysis.
Okay, apologies for not submitting a title in abstract, but I hope this kind of fits. So as we all know, the cost of sequencing has dropped very rapidly, certainly between 2005 and 10, but even over the 10-year period, it dropped by 5 orders of magnitude. And it flattened out a bit in the last few years, but actually the announcements of the X10 and some of the other things people have been hearing about indicate to me that it's going to start dropping by substantially faster than Moore's law again. And so I think there was a lot of innovation actually in the computational methods for handling sequence in this period. And basically we're entering a time where that's going to be needed again. That's, in my view, a key message. So we want fast algorithms, and I would argue that, in fact, for the Sanger Institute, our dominant cost for computing is storage at the moment. And also, as you'll see during the talk, I'm going to argue that more and more, in order to be fast, and in order really to get the most out of data, we want to start analyzing new data in the context of previously seen data, not just a simple reference sequence, but a better representation of all the previously available relevant data. So storage is a critical thing. So we want compressed data. Data compression, I think, is a really important factor along with speed in the way we think about handling sequence data. And I'm going to talk about this whole class of methods based on the Burrows-Wheeler transform, which some of you will be aware of, different levels, I think, around the room. And I've been came across these about 5 years ago with Hung Lee, who developed the BWA algorithm that many people use for mapping reads. And I'm going to talk about a whole series of applications of Burrows-Wheeler based approaches to matching and assembly. These are published, variation detection, and then to trying to represent both read information and haplotype variation across different strains of a species, which are more recent. So this paper just got published a month or so ago. And I basically have been more and more persuaded that this is a really natural data structure and the algorithms that go with it for managing sequence data in general, but in particular biological, with genetic relationships between the sequences. There's some very tight conceptual relationship that I still think is under, it's not all that hard really, it's slightly counterintuitive, but not that hard, and I think it's underused as a representation and as a framework for thinking about analysis methods in our field. So I'm going to try and cover all these things. We'll see how we do. So the basic operation is illustrated here. This is using the Burrows-Wheeler transform and an associated index, which is called the FM index, for matching. And that's what BWA and Bowtie use. And the task here is we've got a reference genome, which is we're representing by Mississippi, by a string of letters. And we're wanting to find matches of some substring into that. And the particular example here is SI, and you'll see that you can find SI here and here. And you could, of course, scan all the way along, but that's a slow process. We don't want to, because this will be billions of characters long. So early on in the 80s, I guess, people came across the concept of a suffix array, which was to take this string and to take all the suffixes, all the things that start at some position in the string, and go to the end, and they're all written here, and sort them. And we have the dollar, we put a dollar on the end as a sort of end symbol. And so they're all the I's come here, and then M and P, and then all the S's. And there's one suffix for each position in the string, and the suffix array, this number is just where in the string it comes from. So Mississippi, the full word starts at position 0, Mississippi starts at 1, Sissippi at 2, and so on. Okay, so why do all that? Well, this is like making a telephone directory. Obviously, once you have a sorted structure like this, then you can look up. So SI, you just look at all the, they're in sorted order. You can just look for where SI is. And in fact, in general, there's a set of matches. It's not just one. There's 2 examples here. And the set of matches is going to be defined by an interval in the suffix array. And so we can have a lower bound of the interval and an upper bound. And I'm defining the lower bound as inclusive and the upper bound as exclusive. So the difference between them is the number of matches. We have 2 matches. Okay. So that's Gene Myers and Mamba and other people. And these are related to suffix trees, which are data structures which people had studied for some time in the 70s and 80s, and there's a variety of machinery based on that. So the insight that Ferragina and Mancini came up with is that you could, if you think about things this way, with having a lower and upper bound, you can have an updating rule that starts with an empty string and updates these as you extend the search string. And that updating rule is going to depend just upon it's going to be linear in the number of elements in the pattern, not in the size of the string. So here's an operation. The empty string will match anything. So that's how you initiate things. And then we have an I. So that's the first character, and that's an interval from here to here. And then we're going to prepend, put the next character on the front, so we have SI matching from 8 to 10. And we're going to have a constant time update for this way of updating these 2 locations in this array. And you can see they jump around. It's not a narrowing down. You started with the whole thing, it jumped to here, and then this interval, the next step, jumped to here. And if you had SSI, it would jump to an interval down here. So, okay. So what is the updating rule? Well, that was the contribution of the Ferragina and Manzini paper. And they said if you already matched a string, you got an LS and US for some string S, we're going to put a symbol A, a character in front of it. That would be like having the, adding an S in front, one of these symbols. It's just a lookup in 2 different arrays. The first array depends purely on the character you're adding, and the second one depends on that character and where you were before. And so if we have those tables, we could conceptually have those tables pre-calculated, and we just do a lookup. And that lookup is clearly a fast operation, and that'll give us the new value. And then as we want to match, progressively refine the match, and this could be across billions of characters, we just carry on these constant time lookup operations. So we're now matching in a way that's independent of the length of the string here. But we do have this big table to think about it. So we've kind of traded off speed for space. We've pre-calculated these tables. Okay, so this table looks a bit scary. L of S could be any value in the whole length of the string, and it's basically the size of the length of the string times times the size of the alphabet. So what are these values? Well, I don't know if I dare try and explain it. It's actually one of these things that takes most people an hour or so of thinking about, or some number of minutes. Hard to explain in a lecture, but actually not really hard. It's a good exercise to go away for those who haven't done this before. It's exactly what it needs to be in order for the answer to work out. That's the right, slightly circular definition, as many things are in mathematics. But there's nothing really tricky about it. So obviously, any string that starts with an A is going to have to start. So C of A is the number of letters less than A in the target string. So let's say our A was an S. We've got all these I's and P's. And clearly anything that starts with a lowercase s is going to have to come after all of those. And C of A just says where the block of s's start from. So that gets you in the right block. And then P, A at ls, says it's the number of times that an A occurred in this, we're going to have this funny thing here. This is the Burrows-Wheeler transform. We've got this sorted string, sorted suffix array, set of suffixes, and for each one we write the preceding letter. So the thing before a dollar is I, the thing before I dollar is a P, before IPPI is an S, before Mississippi is an M. So this is a permutation of the original letters. We're taking, before each suffix, we're just going to write which are, and they're sorted, we're going to write the preceding letter. And then we're going to ask, when we're moving from the block of I's to the block of SI's, we're going to ask how often was there an S in that list, because that's how far down the block of S's we want to go to. So I think I'll stop there, but it's quite an easy, it's kind of natural and easy thing to do. So this thing here, this permuted set of values, this is the Burrows-Wheeler transform. It's the preceding letter to the sorted set of suffixes. And it's a permutation of the original string, and you can invert it. You can create it by sorting all the suffixes, or there's some faster ways to create it which amount to the same thing essentially, and you can invert it. It's an invertible permutation. Okay. Okay, so how big are these? Well, we can represent C and P in space n. I said it's actually quite big, but in fact we can make it smaller. We've got the Burrows-Wheeler transform. That's a permutation. It's constructible from the suffix tree in order n time. In practice it takes longer, but not much longer. There's people in the room who've worked in algorithms to make Burrows-Wheeler transform rapidly. And this is quite big, but you can subsample it. Because it's derived its relationship to the Burrows-Wheeler transform, you don't actually have to store all the values. You can only you can store the values sampled, say every 128 positions, and then you can fill in the missing values by looking up in the by a short calculation on the Burrows-Wheeler transform. So you can make this actually as small as you want to. You can trade off space and time again. In this case, you're shrinking it a bit and paying a bit in time. But in practice, what one does is calculates the Burrows-Wheeler transform, look how big that is, and then arrange your constants so that this index is the same sort of size. It doesn't dominate. So really we're dominated in size by the size of this permutation. So it's permutation, it's the same size as the original string. It's just a permuted value of the string. So this index is the FM index. So in practice, given a query or a set of queries, this finds exact matches. It finds all the exact matches rapidly. Now, we're interested in typically when we're matching in non-exact matches, either because there are errors or because the sequence we have has got variation, it's different. So, but we can treat these as seeds for extension. That's the way, in fact, that all matching algorithms have worked since the 80s anyway, that FastA and BLAST and all these fast methods, all the hashing methods. So we use this process to find seeds, and then we can dynamically extend them in various ways. So there's standard machinery in here, and that really matters in the practical implementation, but the key operation is finding the seeds. So why do this rather than use hashes? Well, there's actually no inherent gain, except for this fact that a big win is there's no penalty for repetitive sequence in this situation. What we just use is just a bigger interval in the index. The algorithm doesn't slow down because of repetitive sequence. And we can choose how long the seeds are. We can dynamically choose how long the seeds are. So where they are less unique, we can require them to be longer. So BWA matches a query prefix trie, another data structure, to this index. And it allows mismatches and indels and keeps suboptimal stats. And there's newer versions, Hung's written, that are subsequence matches. So they're basically using this concept. And Bowtie and SOAP do the same thing, and a range of other programs. So that's about search. You can use the BWT for search, but the other thing you can use it for is compression. And actually, this is what Burrows and Wheeler introduced the transform for. They didn't realize the relationship to search and matching. They introduced the Burrows-Wheeler transform in order to compress repetitive sequences, and actually that's the basis of bzip. So the bzip compression tool, which people can use instead of gzip, which is a state-of-the-art compression tool, essentially gets its power from the Burrows-Wheeler compression. So how do they do that? Well, essentially you run lengthening code, the BWT, and that naturally compresses repeats. And we can see that with our example here. In places where there's a string that you get many times, like SSISS, then you get a block. Let's see, the SSI and SSI are both preceded by I's. So if there were lots of copies of SSI in the genome, they'd all be preceded by an I, so you get a big block of I's. So if there's one sequence which there are many copies of, you'll get consecutive blocks of characters here, because essentially the value, the SSI value here, which are all sorted alphabetically, is predictive of the preceding value. And prediction gives you compression. So it's that run length encoding. Now, the same property of run length encoding that we have here that gives you compression also in fact speeds up the FM search process. So remarkably enough, if you compress your index, you in fact speed up the matching process, because this business of that we traded off a bit of time for space can take advantage of it. So we've ended up with a compressed data structure which we can directly search, a self-indexed data structure. So kind of interestingly also, BWA and all the matching programs don't use this compression, because actually reference genomes, they're designed for matching to a reference genome. Reference genomes are more or less unique. They don't have much repetitive sequence. And that's kind of essential, because otherwise you couldn't match short reads into them. So we'll come back to reference genomes. I think reference genomes, we should change our concept of reference genomes. But anyway, they don't really use that. It's an extra piece of complexity of code that isn't worth it. It would only give you a minor improvement on space. But once we move to metagenomic data, or to primary shotgun read data, or population sequence sets, where we've sequenced many different organisms, we heard this morning about all these viruses with slight variation, and that's the world in which many people in this room are dealing with, then you have very large redundancy. You have large numbers of amounts of data, either raw reads or processed data, which is redundant, where you have there are many bacteria all with very similar genomes. And so then there's a lot of repetition, and this sort of compression becomes very valuable. So one example of that is in assembly. So instead of taking the FM index of the reference, what happens if we take the FM index of all the reads? You might think, well, that's a compressed data structure of the reads. It's good. We can start looking for matches in it. And that's, basically assembly involves looking for matches. So at a naive level, that sounds good. But actually, the value of making the FM index of the reads is stronger than that. It turns out the FM index of the reads, the Burrows-Wheeler transform and FM index, are closely related to an assembly graph. And that's because, in some sense, they're giving you one base extension information. The whole thing about the BWT is it's telling you something about the preceding position, one base extensions. So just as with De Bruijn graphs, you consider one base extensions, overlapping k-mers, here we're actually dealing with taking a string of arbitrary length and asking what happens when you extend it by one base. Oops, so where am I? Okay. So, so I'm going to show you, I'm going to give you another bit more information. You can compute all the non-transitive overlap structure of the reads, and hence the string graph unitigs, which are the data structure that Gene Myers and Mihai kind of introduced, the overlap graph structure, in order n time from the set of reads. This is assuming perfect reads at the moment without errors. And we also actually encode all the De Bruijn graphs for k up to the read length for any k. So there's a succinct representation of all De Bruijn information in the FM index. And that's the basis of the SGA assembler from Jared, and as well as giving these things, the compression of the Burrows-Wheeler transform means that you can work in memory space that's smaller than the size of all the read set, because you're using the redundancy in the reads. Okay. So there's a kind of key question as to whether the Myers or the string graph is the same as the De Bruijn graph after perfect read threading. Maybe I shouldn't have put that in here. Mihai is probably a better person to address that than me.
I think it's open.
So I'm correct, it's open. Okay. So, so this is slightly backwards in the previous slide. Traditional capillary sequence assemblers were based on overlaps. A long time ago, the Staden and Fratt methods, the graph-based. But finding overlaps in a naive way, if you have to kind of look for all pairs of in a naive way, match reads against each other is an order n squared operation. Or at least it depends upon the depth if you're using some hash scheme squared. And short read assemblers are typically based on De Bruijn graphs. But in a De Bruijn graph repeats longer than k, or k-mer require post-processing. So the operation which people use on an overlap graph is to form a transitive reduction of the overlap graph. And that's to say, if you've got these three reads, for example, then R1 overlaps R2, and R2 R3, and R1, of course, overlaps R3 as well. Then what they do is actually remove the R1 to R3. They say that's already represented, because we've got R1 to R2 and R2 to R3. So we can remove that, and we get a sort of stepwise step of next overlapping read. And that's the basic operation in an overlap assembler. But we can do that on the FM index. We can essentially do that without going into the direct representation of what all the reads are. We can do it by looking at these arbitrary k-mers, looking at these k-mers which are the overlaps, and looking for what is there that extends me by one base, or by the next extension. And we can generate this the non-transitive predecessor successor nodes without finding all the overlaps. So that's it's a linear time algorithm on the number of reads. And in fact, what SGA does is it goes straight to what are called the unitigs. It instead of so all the things which overlap uniquely and where there's no split in the graph get collapsed down into single nodes, and this reduces memory by a large amount, and then it fits into memory, and there's no loss of information in that. So this is all prior to these sort of pruning operations which we heard about this morning. Okay. So there's some additional steps. In some ways, that's all very nice and elegant, but actually, it's only the sort of core piece of an assembly. Just as the finding the initial seeds is the core piece of a matching program, there's a lot of stuff around it. There's also, it's just the core piece of an assembly. And practical assembly algorithms deal with all sorts of other issues. And the key operations, as far as I'm concerned here, are error correction and scaffolding, using read pairs. We heard a bit about scaffolding this morning in various ways, or using read pairs. So we do that after we've done our primary assembly on the reads, using by mapping them back with BWA and building us some sort of scaffold graph. I think that's fairly standard, the way that Jared did that. The error correction basically means that this is taking a single read and you're looking along it, and you're asking, are there other reads that confirm, that match? And what happens is at this position, suddenly, there are no other reads that match this read. So we've got a high depth shotgun of an organism. This is a standard diploid, well-behaved organism. This isn't metagenomic data, and there clearly are issues about error correction in metagenomic data. But in this case, where you get such a drop, you can identify, oh, there's probably an error here, because it's probably, or the depth suddenly drops to a low value. It might be there's another read with the same error, but there won't be very many of them. And then you can search around and find an alternate edit, which restores the high coverage. And you can do this in a way that's sensitive to the quality scores, so that it makes use of other information about how confident you are in the read. So essentially all assemblers, or most assemblers, use these sorts of methods so as to get more accurate read set to start with, and then they do assembly, and then they have subsequent processes which clean up other sources of error. So I was talking to Mihai before. In some sense, this error correction is quite closely related to assembly, and for me to sort of say, oh, well, once we've error corrected all the reads, then we can assemble well, is a slight cop-out. But it's one of the nice things about the current Illumina sequencing data is it's really very accurate these days, and the majority of 100 base reads, substantial majority, are perfect. And so, identifying the good reads one way or another, we don't even have to correct them. We could just throw out the bad ones. There's enough good reads in many cases to just work with those. So this is a sort of indication of its performance. This is on a whole human genome, 1.2 billion reads, 120 gigabyte bases of raw data, 40x. There's lots of steps, as you can see: indexing, correcting, re-indexing, filtering. The key things, though, are merging and assembling, and those those work in 48 gigs of memory, and the whole operation works in 54 gigs of memory, less than half the size of the total data. And substantially different from most other short read assemblers, which work in hundreds of gigs of memory to assemble a 120 gigabyte read set. So it's compact. And this is also pretty fast. Most of these other steps take more time. You can see indexing actually is the most demanding time. In the assemblathon competitions, this did quite well in both of them. I take some exception to what, the comment that a single value can summarize the performance of an assembler. I think it does matter. As Mihai said, what you'll care about. The property we liked about SGA was that it actually, although it didn't give the longest N50, it had the fewest substitutions and the second fewest misassembly errors. And we were looking to have a high accuracy assembler because I'm really interested in variation, and I'm interested in not losing variation. I'm not so interested in and accurately determining alternate versions of the sequence. I want to keep my bubbles. People in assembly want to get rid of bubbles, but bubbles actually can represent variation, and you want to keep them. And so we were very happy with that as a performance. So to move on to variation, how do people normally study genetic variation? So we've already seen how the Burrows-Wheeler transform, as well as being a compressed data structure and giving you fast matching, it has this innate relationship to the way that sets of sequences overlap each other and are related to each other in assembly. So the standard way that we find reference variation is to get shotgun sequencing reads, map them to a reference genome. There are problems with mapping. There are problems with repetitive areas of the genome, and you can get local alignment problems with mapping. And then to detect variance based on the multiple alignment of reads. And there are statistical issues you could address here to do with errors and sampling and so on, as well as with these alignment types of things. And just illustrative, I'm basically actually don't work on metagenomics. I'm here slightly under false pretenses, possibly. But I've been working mainly with human genome and vertebrate genome sequence. This is in the 1,000 Genomes Project. These are variants found in two different populations who are essentially the same. There's a British sample of 100 individuals, and a sample from Utah, who are essentially of Northern European ancestry. And we should find that a variant present in Utah is also present in Britain if it's a common variant. So this is going to higher allele frequency 1 here and 0 here. But we found a set of variants in an early version, this is quite a long time ago, a few years ago, that seem to be present in the British but not in the Utah. So that doesn't seem very plausible. And if you look at where they are in the genome, then they're all stacked up around the centromere and the telomere, and in very non-uniformly across the genome, very repetitive parts of the genome. And they're due to problems in mapping to repetitive areas. And there's about 1% of the total that you can remove with some quality filters and sort of post-processing, which actually get rid of these sorts of effects. So now another sort of error that one gets is if the variant is sufficiently distant from the original sequence, then of course the mapping programs don't map very well there, and so it's hard to find those variants. And an example here is where there's that's an alignment error. Here there's an indel. Well, here this is the original alignment. These are all potentially, you might say, oh, we've got a T and a G and a G here. We've got all these variants and a C here. But actually, if you look at the top, this is a very repetitive sequence. There's 4 T's CT and 4 T's CT, so there's a sort of duplicated sequence here. And it turns out that the correct answer is that there was a deletion at this position, and that explained all of these apparent mismatches are explained by a single deletion event, and the reads misaligning across that. So an alignment-based approach here would give a cluster of apparent, in fact, not slightly anomalously behaving variants, but it's a much cleaner version. So, there are other approaches to this, but the real reason is that we're mapping to the wrong reference, and that inherently mapping to a reference is the wrong thing to do. I mean, if you're interested in exactly what isn't the reference, then you're kind of, and all the methodology you're using for mapping is all based on thinking that the reads that you've got come from that reference. You're kind of most interested in the places where your whole framework for analysis is false. And so, I would like to get away from using the reference when we're trying to study genetic variation. And so, reasons why we might want to study use reference-free methods to discover variation, particularly are important when we want to consider variants between two copies of the genome which are quite similar to each other but might be quite different from the reference. So an example is if you have a family, two mothers, or a father, a mother, and a child, two parents and a child, and you want to find de novo mutations in the child, and this is important, both medical genetics as well as population genetics. It's a bit daft. It's a bit like measuring, the width of my hair by measuring to the end of the room, and then from one side of the hair and from the other side of the hair and taking the difference, comparing to a reference sequence. It's clear the child is going to be very like the parents' copies of the genome, the ones that it inherited, with very small differences, 1 in 10 to the 8 or so. And so, rather than the 1 in 1,000 difference there is to the reference genome, there's a 10 to the 5 difference in scale. So you want to compare the child to the parents, not to the reference. Somatic mutations in cancer, you want to compare the tumor directly to the normal. You don't really want to compare the normal to the reference and the tumor to the reference and then contrast those. And even when you're looking at population variation, you want to identify segregating sequences that are different amongst the individuals rather than to a reference. Okay, so we could assemble each individual first and then compare the contigs. So that's one way to do it. But you need high depth sequence to assemble, and it's time consuming. And really we're only interested in the differences. We don't want to go through all the work of assembling the stuff that's all the same. So using these kind of assembly FM index based methods and k-mers, we can start by looking for example, a k-mer which is present only in the child but not in the parents, or only in the tumor, let's say, not in the normal. And then we can extend that outwards until we find a sequence present in both the tumor and the normal. Okay, those ones here. And then we can extend those out in a sort of assembly frame to build an alternate. And we can essentially generate local little haplotype assemblies of the variant against the reference. And that's something which we can do in order the number of variants rather than order the size of the genome and the entire data set. Okay. So what we do is we do this and we evaluate it with this Dindel program which Kees Albers and others developed for it's a Bayesian analysis of evidence, basically using the quality scores and other information whether an alternate haplotype is valid. So some of you may have seen this before. This is with two yeast strains co-assembled. You get a little pattern like this where the grey are regions which have been identical are present in both, and the blue and red are present in only one sequence or the other. And the number inside is the length, and we're using 100 base, I think, 100 base reads. So here we have a singleton at this position, single base difference, and we pick that up. And here we have a long indel, and it looks just the same in an assembly, a variation graph, because it's still just an alternate bubble. But a standard alignment-based method wouldn't reconstruct the whole of this, because you wouldn't align your read properly across there. So really this does allow us to pick up alternate versions. And for example, in the 1000 Genomes project, here's an example where there's a 4-base change. There are 3 reads actually in the standard alignment, so it was picked up, but when we do the variation-based approach, we pick up there are 43 reads. So there's 40 reads which either don't map anywhere in the genome or map somewhere else incorrectly, which actually belong here. And we correctly assign them to, so we pick up the correct number of individuals and the allele frequency for that. So we've used that in the 1000 Genomes recently in a sort of regional form. But that's still somewhat reference-based, actually. I mean, that's still kind of... So how far can we go in taking this? So we're interested in asking how far we can go towards reference-free analysis of data. And I think all this is very relevant to people metagenomics, where you want to kind of work off the big pool of reads and ask questions about alternate versions of particular sequences of interest, like genes or regions. You might pick up one read and say, what else is there that's related to this? What exactly matches it? And what else is there which is comparable, which is an alternate version of it, and build locally. So to do that, you'd like to represent the entire read set efficiently in a way that you could then query and do these sorts of operations on very quickly. So we're aiming to try and build for the 1000 Genomes project a read server that allows queries over the complete set of reads. We can ask questions then like which samples contain a given haplotype, which reads support a haplotype. We can provide a local assembly across all samples around some starting sequence. And we can also just look for new strings that aren't present in the reference or in any previous annotation, which have sufficient support to define new variants. And then we can decide where they belong on the genome after we've found them, rather than doing it all based on the genome. And so to support this, we propose to build a full-text searchable index of all the reads. And we want rapid return of all the reads containing an arbitrary k-mer, and a secondary index which tells us which read is in which sample. So we're going to throw away the information. Okay, so what are the challenges in doing this? So we've got 2,500 individuals at 6x, about 45 terabases of data. I'm building an index over that. Google and so on do this. So obviously, the world can handle these things, and we in genomics should think about it. So we're going to use a run-length encoded BWT to compress redundancy. And what we're going to do is there are going to be many exact duplicates. Whenever there's something is a common haplotype, a common sequence, there's about 15,000-fold coverage. There'll be about 150 read starts for every base pair. So we're just going to store that read once, so that there'll be the same read 150 times or so, if it's a common read. And then, of course, there are places where there's a genetic variant. But those ones are rare. They're only going to be present a few times. Typically, in fact, most of those are singletons. They're dominated by the very rare things. So less than 10% of variant bases are common, actually, that should be. So as long as, the genetic variants actually won't add all that much to this structure. You can calculate it, that you're dominated by this, and the issue is we need to remove errors. If we have a 1% error rate, then we're going to be dominated totally by errors. So we're going to want to error correct first. The proposed solution is to error correct the reads. We only correct bases less than Q20, and we use a cortex structure. We heard a bit about cortex this morning, and if Zam comes, is he here? We'll hear about cortex more, I think. And then we build a compressed BWT of all the unique reads, and I want to keep it in memory, and there were some ideas about improving the compression. And then we build a secondary index then of which samples a read is present in. So we just ask, the first question we can ask is this read present, and can we extend it, and can we match to it one of these searchable indexes, and can we extend it? And then we can secondarily ask which samples, how often is that read seen and in which samples? So we can do these questions across samples. So this is a kind of key value database, and there's a lot of technology for doing this. The key is we're just using the read sequence. We don't have any identifiers here. We lose all the identifiers. We use the read sequence to identify itself. Use that as a key to look up the read count per sample. Okay. We have to do some things because they vary in length, and we're assuming that all reads, we basically clip them to 2 standard sizes, and we quality clip them a bit. So here's a pilot on 1,000 samples on 5 megabases of aligned data. But we just forget about the alignment. We take a region of chromosome 20. So there's 370 million reads, 33 gigabases. It's 1/600th of the genome. That's the BWT of those reads. This is after some clipping and then after some additional compression tricks. And we can get down to 28 million unique reads. And it's in 222 megabytes of data in the BWT, which is under a 100th of a byte per raw base pair of data. So we're really using the compression value of this high highly oversampled data. But we can gain almost all of that back. That's what this tells us. And it extrapolates to 133 gigabytes for the whole genome on 1,100 samples. So that was a pilot done last year. Xicheng Liu and Shane McCarthy are now building a version to actually hold the entire data set. And we think we can build inside a 256 gig memory space the primary BWT. And then we'll have, well, this would be actually allowing multiple ones of them. But we think we can have a scalable solution. We think we can hold all the 1,000 genomes data in one of these. And then we have a set of these level DB key value databases for the other things. And we can retrieve, in reasonable time, we can do these assembly type operations on very big read sets. So I'd argue that these sorts of approaches could well be of interest in metagenomics. Okay. So there's one more thing I want to tell you about, and then I want to kind of try and put it together at the end. So that's one way. This is sort of saying we should go back to our primary data. We should try and get it to be clean. If we have clean primary data, we can work directly with it if we are smart about that. So an alternative approach is to think about sequences that come from the same species, and they're related by an ancestral tree, and they differ due to mutation and recombination. And this is the situation we face in human genetics. We have thousands, and we're going to soon have millions of human genome sequences. In fact, at the haplotype level, we're currently genotyping the UK Biobank. There's 500,000 people in it. There'll be a million haplotypes, essentially, of genotype data later this year. So consider them as a set of aligned sequences with variants at a subset of sites. So you've got some sort of matrix, individuals on one as rows and sites as columns. And that framework is how a lot of human genetics operates. You're interested in questions across sites, across individuals at particular sites, and doing tests and analyses on those. And a key motivation for what I'm going to talk about is the task of imputation. And this is what that means and how it's used in practice. So we might have a set of sequences, reference sequences, from, say, the 1,000 Genomes Project, or this is the 10,000 UK samples. And then you might have a bigger set of samples from which we have partial data. And they've been typed at a subset of locations shown here. So these particular columns, you've got partial information. You might also have partial information because you've got low coverage information or weak sequencing information, which is is incomplete, where there's sampling incompleteness. So the question people want to do is, what can we say about these individuals? Can we say anything about these individual here, this one who's diploid, who I've got this partial information for, what their genotype is here? Can I say something about their full genome sequences? And the answer is yes, because those individuals all come from the same population which is represented in this sample. And the way you can think about it is coloring these separate haplotypes, separate sequences here with separate colors. That's your reference. And what we do is we look in our other set of genotyped individuals with partial information for matches to the individuals, to the reference samples. So this bit here, this GTAA, matches this purple section here. And once we've identified good matches, then we can fill in the missing data. We copy down. And we heard this morning actually about these sorts of methods also for viral sequences with recombination. And in some sense, you're allowing recombination here. You're allowing this to be represented, this sequence of this individual to be represented in part by that reference individual and then in part by this reference individual. The more reference individuals you have, the more likely it is that you have a close relative or a close sequence. And the longer these matches are, the better they are. Okay. So that's imputation, and it's all done in a statistical framework, and this is all quite well worked out. And so this is somewhat, I've made it simple here, but these matches, you can handle these in a probabilistic fashion. Say you might match this one or it might match this one, and you can weight those matches in a Bayesian fully correct fashion. And these things are, not incredibly slow. They use hidden Markov models, and they have a sort of select and copy mechanism. There's a program Impute and Mac. Actually, Daniel's been involved in these things. Other people in the room probably. But But they aren't that fast either. They're quadratic methods. They use MCMC typically. They take thousands. In fact, they're now the dominant use of computers at the Sanger Institute are on these sorts of algorithms, no longer on the matching algorithms, which are very fast and computationally fast. These are all, I like to think they're a bit like using Hammer or BLAST. You know, people don't use BLAST for reads anymore. They use much faster algorithms, although there was a theory behind BLAST. And similarly, there's a strong theory here, statistical theory, but they're not going to scale to millions of samples. The field that's working on them is working on ways to speed them up, but I think they're going at it from that side. So it turns out there is a Burrows-Wheeler type version of this haplotype matching problem that we can use to dramatically scale up these these sorts of methods, these matching methods. And that's, I call the positional Burrows-Wheeler transform. And the reason why it's positional is we've got aligned sequences. We know the standard Burrows-Wheeler transform and the matching algorithm has a reference, and it's looking for a subsequence somewhere in that reference. It doesn't know where, and you're conceptually sliding it along. Here we're not sliding. The problem isn't sliding a new individual along here because we know which location we're typing, but the problem is that there's a very large number in this dimension, and it's trying to find local matches in a big space. So let's imagine, though, that we'd sorted all our sequences. We're looking at one position, k. This is the kth position from left to right. And we've got all these sequences not in a random and arbitrary order, not in the order they first came to us, but we sorted them. In fact, I've sorted them in a backwards direction. So we've got 0, 0, 1 here, and then this is 0, 1, and these are all zeros, and then we've got 2 1s at the bottom. And so these are in standard numerical sort order, but running backwards. And the red things are the length that's shared to the previous sequence. So once you've sorted things, then of course matching is fast. We've seen that right at the start. So we can now look for, if you have a new sequence, if you just throw it into this sort order, if it went here, you'd say, oh, okay, the thing I match best, looking leftwards at this point, is the one that's above me or below me, right next to me. So if you can somehow keep track of this sort order, and the trick is you want the sort order will be different at each place in the genome. So you might say that's incredibly inefficient. How can I manage all these sort orders which are different at different places in the genome? There's actually a simple way, though, to update the sort order. You can just move from left to right, updating the sort order. And the reason is if you're sorted at this position, then you look at the next position. The next position is shown here, which is the same set of data as before. Oops, sorry. Okay, so we're sorted looking leftwards at this point. This is the position at this value. So if we move one right, and we want to sort including this, of course all the ones with a 1 here go to the bottom, and all the ones with a 0 go to the top. But then once you've taken all these zeros, and they move to the top of the new sort, they're going to stay in the same order, because behind them, they're already sorted. So it's a sort of deshuffling process. So all the ones with a 1 go to the bottom, all the ones with a 0 go to the top. That's a linear process. You just sweep through once, and you get a new, and they're sorted in the right order at the next position. So we can update the sort order in linear time, trivially. It's very fast, and there's a simple notation for what you're doing. So we can sweep through and look at all the sort orders at all positions in one linear pass. Okay, so what does that gain us? Well, it's going to allow us to match, but also it gives us compression. So, so something that happens in this, which you can see here. Okay, we've sorted all these so that they're similar to the left. So you get these blocks of 1s, because if they're similar to the left, and they're made of these haplotype sequences, then that's going to be predictive of the next value. This is just like in standard Burrows-Wheeler compression of strings. But because people tend to share haplotypes, which is the whole basis of imputation and the way that we've inherited them from common ancestors, we'll get blocks. And this sequence of 1s and 0s here isn't random. You're going to get blocks of 1s and big blocks of 0s. And you can see some of it on this scale. But as you go to more samples, that's going to become a much stronger effect. So I introduced a sort of simple run-length encoding, extremely simple. I simulated 100,000 sequences of length 20 megabases with a simulator with Max. And There are 370,000 sites, that's 1 every 50 bases, that would be typical. 37 gigabytes of raw output. So gzip compresses that to a gigabyte. You might think that's quite good. It's 35,000 fold compression. Of course, there are a lot of rare sites which have low frequencies. So there are more zeros than ones in this. But the PBWT, with this run length encoding, compresses it down to 7.7 megabytes. That's 5,000 fold compression. So it's much more compressed. And that's not because the run length encoding is clever. The run length encoding applied to the raw data was significantly worse than gzip. It's because of the reordering. It's because you're sorting it so that things are predictive. And so that compression, of course, gets better the bigger the data set is. As you go from 1,000 up to 100,000, the compression improves. And on real data, you also get compression 6-fold better than gzip on 2,000 chromosomes, and we've now done this with 15,000 chromosomes. It's again, even better. It's not quite as good as the theoretical model, but it's substantially 10-fold better or so. Okay, so what about that's compression? What about searching? Well, the update rule is about similar to that for sorting. Actually, it's published. You can look that up. So we can build indexes like the FM index on top of the PBWT, and it turns out they're very fast, and they make your matching for a new sequence to be essentially independent of the haplotype time. But at the moment, I haven't made them very compressed. At the moment, they're quite memory hungry. So another approach is to match a batch of new sequences, basically to build a new structure with the new sequences alongside the old ones and sweep through the whole thing once. So this is order nm in time, but it's very memory efficient. And if we do that, so I've got here 3 methods. Naive, where I match each sequence in turn to each of the other sequences. That scales, going from 1,000 up to 50,000, that scales linearly, so this is 50 times as long as that. It's not, when it says naive, actually it's quite a clean implementation, I should say. It's efficient matching of one sequence to another at a time. If we use the index data structure, it takes a second, and that's independent of reference panel size. So I can take a new sequence and find the best matches, local best, all the local best matches in a time that's independent of reference panel size, until I run out of memory and I start swapping. This is on my laptop, so and I take 15 seconds of system time here, you can see. But the last of these is the batch method. Basically takes no memory and has no swapping problems, but as you can see, increases slowly, but very slowly, I think in a logarithmic sort of fashion with scale. So we get really big differences in matching time. Okay, so what about inference? That's all about compression and matching. Well, we've used this fact that we're predicting the next value to give us our compression, and we can actually think of a generative model. We can think of this probabilistically as predicting y given the degree of match to the previous sequence. And we can use this as in an inference framework for imputation and phasing and so on. And we've now been implementing this for imputation and sort of exploring different strategies for phasing. So this is some data from a rotation student on the haplotype reference consortium, which is trying to put together large numbers of sequenced genomes. There's about 13,600 samples at the moment, 27,000 haplotypes. Chromosome 20 is 430,000 sites. The blue line shows, so here we're showing imputation accuracy. This is the allele frequency. This is the R squared. R squared should be 1. It's the correlation of your inferred value to the truth. You'd like it to be 1. For common alleles, there's a lot of data and you do well. As you get to rarer and rarer alleles, then there are fewer examples and it's a harder thing to do. So typical imputation performance graphs look like this. The 1,000 Genomes project is the kind of current state of the art that people use in human genetics for imputation. Using impute2, and that's this blue line. You can impute down to 5% or 1% moderately well. You'd like to impute to 80.8 value, so The black line is using the haplotype reference consortium with Impute, and the red line is with PBWT. So we actually outperform Impute now. I was talking to Jonathan Marchini, the author of Impute, and we think we understand why. But as well as doing it, outperforming it, we took 55 seconds to do that. Impute was running to do it on one sweep through the whole genome in 87 megabytes of memory. Impute used 8 gigabytes, and basically, well, it did it in pieces, but the total time was 38 minutes. So there's a major difference in space and time, essentially. So, okay. So, to finish up, I think that we're really taking advantage of the shared data, of the shared sequence structure, essentially, and of these computational representations and algorithms that, by using sorting structures, give us fast and compact ways to manage shared sequences. I'm convinced that they're good for all sorts of things in the field. I think it's really worth people who want to use large amounts of sequence data, learning and understanding and write algorithms about them, these data structures. Kind of a big open question is, can we merge these PBWT ways of thinking about things, where you're looking about variation at defined points across a set of individuals, and the assembly sort of raw sequence read server type way of thinking about it. But the sort of large-scale vision is that we'd like to build a model from all existing primary data to use efficiently for interpreting new data. And I think we're starting to, there's a new organization, the Global Alliance for Genomics and Health. I think we should start thinking about reference representations, which aren't just like a single E. coli sequence, which everything gets mapped to. We know that there are strains with all sorts of extra sequence. Nor should we think about something like the current human reference actually has alternate haplotypes and extra pieces, which are sort of little blocks. We should try and represent what we know about the variation and then map into that, so that we don't keep on spending most of our time finding things that have been found before. When we sequence a new strain, almost everything that we see has been found by somebody else before. And if we knew how to look for that already and had it represented in our matching process, we'd match straight into that. And then we'd know which version of what's been seen before we have. We could probably make do with much weaker data. You could kind of say, yes, I've got enough evidence to verify this thing I've seen before. I don't need to discover it all over again from scratch. And then we can focus on the new things, which are really different. So can we move away from limited linear references, combine a computationally efficient representation with probabilistic models for genetic structure? We're going to want to move to distributed data sets in the longer term, so that, because having everything in NCBI, well, even now, there was a time that we've oscillated back over the years. I remember back to the 80s, there was a time when GenBank, people used to download it every night, and you had a complete copy of GenBank, and people searched their local copy. And then, of course, GenBank introduced BLAST, and people searched there with BLAST. And then, now with new tech reads, people do their matching and searching locally again. It's not true that NCBI do all your mapping for you. But there's this kind of oscillation of doing things locally or distantly. And it has to do with technology largely, as to trade-offs between whether they can afford to map for you. And I think we need ways of representing more of what there is out there than the current set of references that we map to. We need to represent better in our mapping process the known knowledge. So that's where it ends. And the key people are Hung Lee for the early steps. And in fact, Hung has written an assembler, as you probably know, Fermi, that uses the Burroughs-Wheeler FM structure. Jared wrote SGA. Vladimir is a very smart mathematician who's interested in PBWT structures, and John did the later the imputation analysis. And Shane and actually Ji Chang, who isn't on here, are involved in a lot of handling of large data and being key people. And these are all people who have contributed to big data sets that have been the motivation for this. So thanks.
So firstly a comment. I think the 1,000 genome read server sounds amazing. And for metagenomics, certainly for my research, having something like that to interrogate some of the large data sets that are out there would be a real boon. So I'm quite excited about that idea. And the question was, on the PWT for haplotype reconstruction for metagenomics data sets, could that work given that bacterial genomes are not in LD and it's a bit of a different issue with replication?
So it's not, I'm not so sure. It depends a lot. I mean, it's looking for shared, it's looking to glue together pieces of shared haplotype structure seen in other individuals. And I think one of the things is all my individuals, it's not, I'm assuming that I've already got, well, there's a phasing problem actually. The problem of separating out the haplotypes, which are all mixed up in your sample, in humans is just you're going from diploid to haploid. That's what we call phasing. That's a much bigger problem if you have a big mixture of different haplotypes. But given a reference panel of things, essentially this allows you to glue together pieces to explain something new in terms of pieces of older things. So I think that could carry across, and yeah, I think there could be something, but some of you know more about that.
There's certainly haplotype structure in bacterial genomes that can be exploited.
It's going to depend upon the bacterial clade, actually, I think.
But yeah, in a big assortment of metagenomic data, there's going to be lots of different models of evolution in there, aren't there?
Yeah.
Yeah. I think this uses well the kind of where you have pieces that have recombined across from each other. I think that this will use that structure.
You haven't tried it yet?
No, not on that.
So once you have the index, searching is really fast.
Mm-hmm.
But how long does it take to build the index, and how often do you want to update it?
Yeah.
Or do you have to rebuild it actually, or can you update it?
Yeah, you can merge them. There are processes for merging them. One of the ways to build some of the class of algorithms for building the indexes. So Tony, who's behind you, has been involved in some of these things, involved making indexes on subsections and then merging the separate ways, the different ways of kind of slicing and dicing the building process. I think it is an important question, making these structures updatable, actually. You can also kind of, at linear cost, you can have a big one and a second one, and you can look up in both, and you can put some machinery on top to manage a sort of serial thing and then bulk updating. But I think this is a world, again, I imagine that this is what, the Googles and search index people address all these issues about secondary and, having transitional processes for updating indexes. So I think a lot of that will transfer across potentially. So, but the action of introducing one new sequence into an index is pretty heavy. You don't want to do it one by one.
But your imputation algorithm does have a statistical component in it, does it?
Actually, the thing that works best is just to look for all the maximal matches that, and yeah, there's some kind of rationalized. So I'm a little skeptical. The argument that Jonathan and Peter and people make, that it's all very well-founded statistical approximation to the coalescent, I think is a bit bogus. I think it's slightly post hoc. They have a probabilistic model, but I think its relationship to the true coalescent structure is a bit tenuous. So yeah, you can give it a probabilistic interpretation.
I mean, you have to, because you have the forward and the backward, and you have to decide how to integrate them into coming to an answer.
You can run it forwards and backwards. This thing, yeah.
Okay, but how?
You can also run it one way direction with traceback.
The process you're doing, is it deterministic or is it actually?
The way that I impute at the moment is deterministic, but you could infer it as an optimizing something. I could talk to you afterwards.
So
I've tried different strategies actually. So.
we'll take one more question and then we'll have the coffee break. Is there another question? Wally?
So I don't know much about this, and so my question is probably not well posed, but as I understand it, with the first thing that people want to do when they get a new sample of metagenomic sequence is to map it to some set of references which happen to exist already. And of course you get lots of possible sites of match in this, and what happens immediately is your data set becomes 100 times bigger. So it's already too big to upload to some of the servers, and then it gets, the whole thing becomes such a complicated thing, and it's all to do with data storage, data science. So I mean, it looks to me like this is going to be potentially very useful. First of all, we could sort of reduce the reference databases using this, and then hopefully we could reduce the samples in the same way, and then sort of bang the two together somehow. I'm not sure if I got that bit right, but it seems to me that one could actually gain in many ways from this. But the key is, I think, whether you can take your compressed data and match it to the compressed reference database. Is that going to work?
Yeah, so I think this business of having a reference database which has got a lot of references that are so similar to each other that your sequence is matching many of them, and then that's kind of central to this, and it is a bit pointless to keep then all these separate matches when they are the same thing, essentially. So interestingly, if you use BWA on this set of references that all look the same as each other, well, it doesn't compress them. If you used an FM index matcher which uses a compressed reference set, then it would naturally account for that. It would shrink down where you had the same thing repeated. I don't think that all the pieces are there to do exactly what you would want right now, but I think that this machinery, I think the core things are present, and that you'd like to think about, representing once that interval basically, rather than splitting it all out again after you've done the matching. So yeah, but I haven't really, I haven't engaged with the metagenomic data.
Okay, thank you very much.